Hello, my name is Muhammad Noor Alif. You can call me Alif. And I am from class 119 PDT.
Hi, I'm Rashida Nurulisa from 119 Teacher. Hi, my name is SABRINA. You can call me Sak. I'm from P1M1.
Hi, my name is FATINI. You can call me FATINI. I am from class 119.
So today we'll talk about recombinant DNA technology. Firstly, the definition of recombinant DNA technology is a method in which DNA molecule made in vitro with segments from different sources or by combining genes from different organisms. Secondly, tool used in recombinant DNA. Firstly is target DNA or gene of interest. Secondly, DNA cloning vector. Thirdly, host cell. Fourth, modifying enzyme. And fifth, restriction enzyme. Okay, for the first tool is target DNA or gene of interest. It is a sequence of DNA containing gene of interest. This is nucleus and there's a DNA inside of it. So there's a line of DNA will be cut and a part of it is used in the gene of interest. For example, gene encode for protein which can dissolve blood clot.
The second tool is restriction enzyme. Enzyme which cut DNA molecule at a restricted site and produce short restricted fragments. Restriction site is the short base pair sequence of 4 to 8 base pair, recognized by a particular restriction enzyme. Restriction site contains palindromic C where the same sequence of base is found on both DNA strands but in opposite direction. So we have EcoR1 and SMA1 for example. Imagine that EcoR1 and SMA1 are scissors. So this is the DNA sequence. So this is the first example, EcoR1. EcoR1 is produced by E. coli. Recognize the base sequence of GAA-TTC. Makes the staggered cut within the sequence. So the EcoR1 will cut the sequence between A and G and G and C. And then it will produce TPN strand. So this is easy for the next sequence to join. The second example is SMA1. SMA1 is produced by the Serratia marcescens, recognized by the base sequence of CCCGGG. The cut is made between the G and C or C and G. So this is the SMA1. The SMA1 will cut between the G, C and C and G. It will produce blunt end.
We will explain about the DNA cloning vector. What I mean by DNA cloning vector is the DNA molecule which can carry origin and target gene fragment into a cell and replicate in that cell. Okay, so let's move on for characteristic. For the characteristic is able to accept the foreign DNA in the MCS and able to replicate fairly in the host cell and possess selectable genetic marker. So, for example, is with plasmic par 18, bacteriophage
lambda 201, and cosmic as cos1, and yak p yak. But for today we'll be focused about the plasmid PUC18, which is DNA molecule that can carry foreign DNA into the host cell and replicate there. For the second one is capacity to carry the insertion size, which is 1 into 10 kilobase. Okay, for example, structure for part 18 is for this one is Z, the opposite one is ampicillin resistant.
In modifying enzyme, we have Taq polymerase and DNA ligase. Okay, for the Taq polymerase is the enzyme obtained from the bacterium Thermus aquaticus. Thus, it is heat stable at 95 Celsius, and it can withstand the denaturation step used in the PCR method. PCR stands for polymerase chain reaction. Okay. Lastly, for the DNA ligase. DNA ligase is the sticky ends of DNA strands produced by action of restriction enzymes and joined together by enzyme, which is DNA ligase. Okay. Function of DNA ligase is to join two DNA fragments together during DNA replication. Okay, for the proof it, for the example, we have a sticky end and blue E as a DNA ligase that then
So next is host cell. Host cell is also known as one of the tools that is used in recombinant technology, right? So what is special about host cell? Host cell isn't like any other cells. So there are three special characteristics, which is the first, of course, it was able to receive recombinant plasmid. Right, so how does this thing occur? So this thing occurs actually through a process called transformation. Alright, so through transformation process, the recombinant plasmid will be introduced into host cell. Right, so second characteristic of host cell is able to maintain the structure of recombinant plasmid. So the structure of our recombinant plasmid will not be damaged. So it will maintain from generation to generation. And the last one is host cell is able to amplify. Means it can increase in number, increase in quantity. So from 1, it can increase to maybe 4 and then 8 and more and so on. So as you guys been wondering, what is the cell that commonly used as host cell? Right, so as we know, bacteria is commonly used as a host cell. So that but in lab, in laboratory, we can't get just any kind of bacteria. So we have one specific kind of bacteria we use. So it is called E. coli bacteria.
Okay, today we talk about step in gene cloning. Gene cloning, for the first step is isolation of gene. For the gene cloning, we involve two cells, which is the left one is donor cell and the right one is bacterial cell, which is E. coli bacteria. In donor cell, we have gene of interest, and this in E. coli bacteria we have plasmid and bacterial chromosome. For the isolation is for the donor cell we isolate the gene of interest. For the bacterial cell, we isolate the plasmid.
The second step is cleave or cut. This is plasmid and this is DNA from the donor cell. DNA from the donor cell and plasmid bacterial is cleaved using compatible restriction enzyme. As such, will be cut on this part and it will be like this. For the plasmid, it will be single cut within the leg Z gene, and this is the site of cleaving, producing complementary of sticky end.
For the third step is the insertion. Okay, this is the donor gene and this plasmid. Okay, the donor gene is inserted into the plasmid and by using the DNA ligase. DNA ligase is used to bind the sticky ends together. Two pieces of DNA are joined together, and finally, recombinant of DNA is formed.
The next step is transformation and amplification. So we can see this is the plasmid with the DNA recombination, and this is the plasmid without DNA recombination. So we start with first the DNA with recombination. So this is a small one. The DNA recombination this is the host cell. The DNA with the recombination will introduce to the host cell, and it will also replicate and form. Also, the DNA without DNA recombinant will also introduce to the host cell and then will replicate. Also, we can see there will be a host cell without a plasmid or a plasmid with the recombinant DNA.
Right, next, after we have produced many bacteria, the last step is to identify whether the bacteria contain recombinant plasmid or not. So there are three probabilities here, which is a bacteria that contain recombinant plasmid with a gene of interest, only contain plasmid without gene of interest, and also bacteria without anything. Alright. So first, the method used is we plate the bacteria on agar containing antibiotic ampicillin. So actually here, in both, we have a region which it has ampicillin resistant. Alright, so after we have placed this all bacteria in agar of ampicillin, so the bacteria without plasmid, which is without the ampicillin resistant, will definitely die. Right, so the surviving bacteria will be produced. So the second method is used, whether to find out the plasmid have gene of interest or doesn't have the gene of interest. So we use a sugar that is called X-gal. So this sugar makes the plasmid produce beta galactosidase because it has a region that is called leg Z. Alright, so if the plasmid contain gene of interest, the leg Z region will be will not function functionally. Right, so there are two possibilities, which is plasmid with gene of interest and plasmid without gene of interest. So by using this sugar, which is X-gal, the region which is called leg Z will not function in the plasmid with gene of interest. So in the plasmid without gene of interest, the leg Z region will functional, and it will produce beta galactosidase to hydrolyze the X-gal sugar that we placed. Alright, so the difference, so to find the difference, the plasmid that produce beta galactosidase will be blue in color, and the plasmid with gene of interest will not produce beta galactosidase, so it will turn white in color. And this is our last product.